Professor Gadre, Dr. Bhushan Patwardhan, Dr. Rama Vaidya, other distinguished guests, and my friends. It's a great pleasure and privilege for me to be asked to speak here for the Science Club, and I must express my appreciation to the Center for Complementary and Integrative Health, the Science Club, and the Pune University for the honor they have done me. The subject I have chosen is Ayurveda and modern science. As you heard, I am not professionally an Ayurvedic physician. Some 30 years I was practicing cardiac surgeon. I started learning Ayurveda when I was past 65. So this has the first 7 or 8 years studying the 3 basic texts of Ayurveda, Brihatrayi, writing books. I enjoyed doing that. But as I did that study and writing, it occurred to me there are several areas in Ayurveda which could be subjected to investigation using modern science. I found that the Ayurvedic research in the 20th century mostly, it has been largely through pharmacology, medicinal chemistry, herbal drugs, developing molecular drugs from herbal preparations and so on. This is a very large area, still very active. Started by Ramnath Chopra, incidentally, first part of 20th century. But there is something else which has happened towards the end of the 20th century. That is where molecular biology has come, a new modern biology has come. Now I found that there are opportunities. Modern science could be harnessed to study some ancient ideas, ancient procedures. That looked like a very attractive proposition to me, and I've been busy with that in the last few years. So what I say will be largely about this in the context of the interaction between science and Ayurveda. I'd begin by saying that the interaction started long ago. It is part of the East-West encounter. India has, had contact with a number of other countries, conquerors coming here. Many of them came for conquest. Many came for plunder. Very few came in search of knowledge to India. This is how it started. Now, the results were different. For example, in the area of music, there was not much of effect of the West on Indian music or Indian dances. Relatively immune. But in the area of judiciary, we have completely a Western judicial system. Our criminal procedure code, we are still following all over. There is not much of protest. Willingly accepted. English language, for example. Science and technology, everywhere. Pune University. These are irreversible changes. But in the area of medicine, that is also interesting. Western medicine, when it came to India, largely in the 19th century, practice of medicine. They tried in Calcutta, initial native medical schools they started, because they could not get enough British doctors. So the first year was a common curriculum taken from Western medicine of those days. And then the next 2 years, the Muslim students would be learning Avicenna and texts. Hindu students were taught Charaka and Sushruta. This was done by East India Company. But I have seen the textbooks actually written. Hari Krishan Singh in Chandigarh, he has copies of these. But there was wide protest from Ram Mohan Roy and many others. The Renaissance in Bengal. They wanted this abolished. There should be one medical course. So William Bentinck ordered, and that is how the modern medical school came. So the interaction has been going on for a long time. I just wanted to take more looking at not the practice of medicine. I'm looking at the science interaction. Now here actually it started from the 16th century. interaction between Western science and modern medicine. Now, this started with a remarkable man called Garcia da Orta in the 16th century. He was a Portuguese physician who graduated in medicine from Spain, and he was looking for an opportunity. He was not a rich man, and a friend of his was traveling as the head of the fleet coming to India, so he joined him. And the 36 years he practiced in Goa from 1534, and he was the first European to write a book on the India's medicinal plants. And he was the first to mention Rauwolfia serpentina, which is not mentioned in our Brihatraya. And he was the first to describe cholera, which was new to him. Ayurveda, we have Atisara, Vishujika, but there is no dramatic description as an epidemic which we are familiar with. That's not the way it is described. Here is a classic description what cholera was. 100,000 people dying in a month in Calcutta, that kind of dramatic descriptions. And you see the first description from Garcia. And this classic colloquies on the simples and drugs of India, which was published in 1563, and 57 colloquies. In those days, the style was, popular style was two people having a dialogue. It was presented in that form to make it more intelligible, more attractive. And in this, the dialogue is between Garcia himself and a colleague of his called Ruano. These are the two people taking part in these dialogues, and they refer to plants largely, the habitat of plants, morphology, use in treatment, commercial aspects even are discussed. And descriptions were given vividly, especially cholera, dysentery, diseases which were strange to him. He described in great detail. And he also describes Sarpagandha, Rauwolfia, Pauli cobra, that is the term. But when he describes this, he recommends the use for fever, rheumatism, smallpox, measles, and cholera. But this Pauli cobra in North India, it was used for mental disorders. There is no mention of mental disorders in Garcia's list. The Portuguese physicians and others who were resident in colonies in Sri Lanka, in India, Macau, etc., they did not pay much attention to Garcia's book. But it received immediate attention in Europe. Widely popular, it was translated into French, Italian, and other languages. And English translation came very late. And in this dialogue, Garcia appears as a liberal person. That is, Greek medicine is the basic medicine, but he is willing to accept something which is seen to be effective in other systems of medicine, Arab or Asian. But Ruano, the other, his colleague, he is a conservationist. He would not accept anything except Greek medicine. That is how the dialogue is presented. And in this you will find many Ayurvedic medicinal plants which you see in the Brihadaraya he is using. But there are also many plants which are not mentioned, like Rauwolfia, because the native physicians in Goa, they had a lot of folk practice of medicine. They were using those. They are also mentioned. The next comes the 17th century. That is a Dutch governor of Cochin, Van Reed. He was not a physician. He was a brilliant naval commander. And when he settled in Kochi as the governor, he found something. Yesterday I was telling Dr. Gadre about this. He noticed that people living in Kerala, especially in the Cochin area, he did not think much about their food. He thought it was a very low, nutritious, unattractive food. But he was puzzled that these people were healthy. They were working very hard, physical labor, climbing trees and so on all day, and they were remarkably healthy. How can with this kind of wretched food, these people can be healthy? This was a puzzle. And then he noticed that in their diet, every day they would be taking coriander or dried ginger, cloves, pepper, turmeric, So there is no food where there is not some small part of these spices, and they take it all their lives. So he started wondering, maybe this is what is giving them this health. So he wanted to study spices all along the Malabar coast. Because he was a governor, he could command virtually an army of 100 people, herbalists, native doctors, professors of botany, artists who could draw these. So he had a very large number of people, and he wanted to have a physician who could tell them about this particular plant, what is it used for. Now, no physician was available, and most of the Ayurvedic physicians were high caste people. They would not cooperate with a Westerner in a study. So Van Reed approached the Maharaja of Kochi. He was the governor, so he could tell the Kochi king, I need some help. So he designated a physician called E.T. Achyutan from the Eedava community. They were considered OBCs in those days. But they had plenty of high Sanskrit scholars and physicians. He was the one who gave all the help. This particular plant, it is used for this particular disease. Now that is in this book, the acknowledgements in the beginning, you can see E.T. Achyutan, they have all agreed. So authorship, it is all taken care of in this book. It is a remarkable book, published Hortus Malabaricus, the Garden of Malabar. 30 years of this study, funded by the Dutch East India Company, also supported by the Maharaja of Cochin and the Zamorin of Calicut. So it is a classic publication in Latin, and it was a real, even today it's a treasure to see that. And 740 plants from Kerala, all along from Kanyakumari to Goa, and ethnobotanical information given by all these people, verification of plant names done by 3 people who actually belong to where I live now. I live in Manipal, South Canara. All these 3 people, Ranga Bhatt, Apu Bhatt and Vinayak Pandit, all these 3 people, the plants which are mentioned here find their place in the Ayurvedic Nighantus and give the reference. These were the people. And so this was translated into English and Malayalam only in 1980s by Professor Manilal. This is just a sample of the illustration. Very high quality, black and white. These are classic pictures. You simply cannot improve them. The original plates for these, there are the engravers, he got the engraving done, they are all in British Museum, all the 740. Now these two, essentially they are working on taxonomy, 16th century and 17th century. Now that created very great interest in the taxonomy in European countries, and Portuguese, Netherlands, France, all these, they were colonial powers. They were trying to come to Asia, particularly India, which was known as a land of great wealth. They wanted to come here mainly for plunder and conquest. That is what they were coming for. But they were scared of tropical diseases. They had heard all these rumors about cholera and so on, thousands of people dying. They didn't have means of treating them. They didn't know how to treat them. So there was great interest in these colonialists about tropical diseases, how they treated. That was the main reason for their interest. And they also knew that there is a market in this. Even at that time, if you could make drugs out of it and sell it, they could make a profit. So that also was there. And Linnaeus, the father of taxonomy, he has actually praised, he says, I wish I had seen this earlier. It was such a great classic, Hortus Malabaricus. But these two, the Botanical Survey of India, was the British took interest in this. Out of those, following these, the 200 years after the Van Reed, among the great people who took interest in taxonomy, one was Roxburgh, particularly memorable. He was in Bengal. He wrote a book, Hortus Bengalensis. He never became as famous as Hortus Malabaricus, but he was a person, such a great scientist, a physician who became a botanist, and he was revered by J.C. Bose. In fact, he used to consider his residence as a place of pilgrimage. He was a really, truly a great scientist, Roxburgh. And all over, even in Madras, there is one Sharif who wrote a book on Indian taxonomy. And many, Ainsley in Chennai again. Now in the 19th century, because of the interest is so great, 70% of the drugs of plant origin in the British pharmacopoeia, they were all of Indian origin. And the Botanical Survey, the British, by that time they had established in India, they set up among the surveys, Zoological Survey, Geological Survey. One very important survey was Botanical Survey of India, which gave further impetus to studies in medicinal plants. Now, so all the first view, science taking on Ayurveda was all through the window of taxonomy. Now in 20th century, something new happened. That was Ramnath Chopra. He was appointed the School of Tropical Medicine, Calcutta, which was one of the institutions established by the British. They not only established hospitals, it is not often remembered that they established a number of scientific institutions. School of Tropical Medicine is one of them. Kasauli Institute is another. King Institute, Guindy. Hafkin Institute, Bombay. So there are about 10, 12 institutions they established, all only for scientific research. There is a certain amount of applied. If you look at that, it's surprising. They were also they were of course making vaccines for all the local use. They were to produce something of practical use. At the same time, they were also in charge of investigating local outbreaks of diseases. They were in charge of that. If there was an outbreak of a diarrhea or a viral fever or something, this institute had to go and get samples from there and investigate. So it's very carefully planned. Government gave funding for this. So this is one of those institutes in Calcutta, which is remarkable work. That's not our subject today. But Ramnath Chopra, when he appointed there, he started a totally new activity, pharmacology. He is actually revered as the father of Indian pharmacology. And he did monumental work. In his own, I have taken 2 paragraphs from his book. His objective in starting this to make Indian pharmacology self-supporting by enabling her to utilize locally produced drugs economically under standard laboratory conditions, and to discover remedies from the claims of Ayurvedic, Tibi and other indigenous sources suitable to be employed by the exponents of Western medicine. This is Sir Ramnath Chopra's. And his work involved botanical identification, chemical analysis, pharmacologic studies and clinical trials of a large number of commonly used drugs. And it goes on, extensive studies on the physiologic action of the active components on living tissues in the water bath, nerve muscle preparations and so on, trying the effect, biochemical and biophysical changes brought about on mammalian organisms. Carried out pioneering studies on Drosophila. And his books, The Indigenous Drugs of India and Medicinal and Poisonous Plants of India, in 2 volumes, they became classics. And he is acknowledged as the father of Indian pharmacology. Now it was natural that organic chemistry, natural products chemistry, that came in the wake of Ramnath Chopra's studies. A large volume of work has been done by brilliant scientists. I can only mention a few: Asima Chatterjee, Govindachari, Sukhadev, and many, many others. And these are classic studies. There is no point reading all this, like name work of Govindachari. These are all very well known for this audience. I can tell you, a few years ago, I happened to be in Paris. There was an Indo-French council, and the French pharmaceutical industry once called us for an evening with them. So I asked them, what is the major focus of your R&D today in France, in drugs? Oh, immediately that chairman said, oh, I can tell you easily, that is aging. Aging is our problem. So we work on that. Then he volunteered. He said, I can tell you, we are actually working on a lead given by one of your scientists. So I was surprised. I said, who is that? This is Asima Chatterjee. Because the work that they had done, it is a huge database. The whole world is using that. That we must remember. It's a great work. Now, 21st century, where we are now, the discovery of the double helix, that changed the entire picture. The way we look at organs, we look at human body, we look at health and disease. Everything has been influenced by double helix. Now, there was a time when, say for example, we were medical students, we looked at organ specialization, whether it is kidney or the heart or the liver. Even before that, it was the whole human being. then it came to organs, then it came to cell, nucleus, DNA. It became more and more reductionist. Now this is very true because very recently we were doing some work on a Rasayana on the heart, this organ. Does it have any effect on the heart? Because Amalaki Rasayana has effect on the whole body. So we were looking at various organs. His whole body, so naturally it should have an effect on the brain, on the heart. So that paper, when we did the initial work, it was done in the rat, and we could show very clearly that cardiac hypertrophy, I'll mention that again later, it has a beneficial effect. It has a beneficial effect on ejection fraction, how much blood the left ventricle is pumping. All these we had shown. Effort tolerance improves. Now this, a top journal won't accept it. All very good evidence. Echocardiography is done, ejection fraction is calculated, it's all there. No, it can't be accepted. But we were determined we should publish in that journal. So what is necessary is the mitochondria, the energetics. That is the mechanism for this, the hypertrophy. How do you explain it in terms of this mitochondrial energetics? That is so we did that. It took another 2 years, 4 years. Ultimately it was published in Nature Reports. So this is what is to be accepted as science. Unless it is carried to the DNA level, it is no longer accepted as science, which is a pity, but that is where it is. So Ayurveda naturally, taxonomy, pharmacology, then we finally come to molecular biology. So this is a new perspective of Ayurveda. Ayurveda remains the same, but we look at it through different windows. Now this is Ayurveda and science, they are not strangers, because P.C. Ray, many of you will know, he was the founder of modern chemistry in India, a very great man in Calcutta at that time when the West and East, they encounter. Now, he was a saint. As a bachelor, he lived in a single room in Presidency College. He wrote the history of chemistry in India, which is a classic. Without even going to Britain, the university there gave him a doctorate for his book, a classic book. I hope your library has it, you should read it. Now, P.C. Ray, 600 BC to 800 AD in that book, he calls it the Ayurvedic period in the history of Indian science. He calls it that because Ayurveda is not only the mother of medicine, it is also the mother of chemistry, because according to P.C. Ray, the whole book is mostly about that mercury, rasatantra, that rasa is mercury. Now that, he has of course covered that very extensively in that. But he has even classics in Rasa Shastra he has translated. To that extent he has gone. So that chemistry is the foundation of Indian chemistry, and that he has traced it through. And Ayurveda, as you know very well, that is an integral part of Ayurveda. So he says that it is the mother of chemistry, but we could also add it is the mother of Indian botany. We have so many veterinary science, because we have Ayurveda for plants, Ayurveda for elephants, Ayurveda for horses. There are big texts for these. So veterinary science also, this is the mother. So it is the mother of all these biomedical sciences. So therefore, to do science in Ayurveda, it is not surprising, because they are very close to each other. And what we are trying to do using this molecular approach is the biological basis of Ayurvedic concepts and procedures, and the mechanistic basis of therapeutic effects. Suppose somebody has got arthritis, you give an Ayurvedic medication and you find the swelling reduces, redness reduces, pain reduces. Now what is the mechanism of this? What is the mechanism of action? Now that is also coming in this. You need this molecular approach to study those changes. Immunologic changes happening in the body. These are all modern biology. And this did not receive any attention in the 20th century, partly because we were obsessed with the taxonomy, medicinal plants and drugs, And secondly, the modern tools didn't exist. In the 1950s, we didn't know much about the DNA, gene sequencing, gene expression. We didn't know any of these things. There were no tools to study them. Immunology simply did not exist. In the pathology text which I learned in the medical college, inflammation is the first thing that we learn in pathology. So in the center of the very epicenter where the necrosis is taking place. But as you go more and more peripherally, you will find various types of cells, neutrophils, different types of cells. And at the very periphery, you will see big collections of lymphocytes. In the boy's textbook, he writes, the real soldiers fighting, these are the leukocytes. They are there, right there, dying in large numbers. But in the periphery, these are the phlegmatic spectators. This is what he writes about lymphocytes. Today we realize they are anything but phlegmatic. The whole cell is full of nucleus. There is tremendous activity. We didn't know that. So immunology didn't exist at that time. So today we have all that. So we can study. There is a new opportunity for us. So Ayurvedic biology, essentially, it is biology, but they take the cues from Ayurveda. That's the only difference. It is an emerging discipline which harnesses the tools and methods of molecular biology to investigate the concepts, procedures, and therapeutic effects of Ayurveda. Now, there are many obstacles in this. It's not easy to do this. First of all, identifying concepts, procedures, and mechanistic studies which would lend themselves to experimental work. Because the moment you talk about research using molecular biology and immunology, you are talking about a laboratory. Now, you have to have the problem amenable to be tackled in the experimental laboratory. Like, for example, Panchapura Siddhanta, that is the macrocosm, the universe, microcosm, the human body. There is a homology. It is a fundamental doctrine in Ayurveda. Very important. At every stage it will come. But how do you make it into a concept? You can investigate in the lab. This problem. That's the first problem which you will face. Second, designing experiments and protocols on the basis of descriptions. Because a description of a treatment, for example, maybe only in 2 verses, written 2000 years ago. They cannot write great detailed materials and methods, several pages, which you need for a protocol. So from this very brief description, some of the terms are amenable to controversy. The measures that they have used, people don't agree on that. There are all these problems when you are dealing with an ancient text. But using that, can you make a protocol which not only you can do, but somebody else can follow the same protocol and replicate the same results? Now that is the second problem. Third, creating partnerships between scientists and Vaidyas. This is exceedingly difficult because they talk 2 different languages. There is no eagerness to meet each other. In fact, there is no forum until perhaps now, like you are doing here. There was no forum where Vaidyas and scientists, they had a common forum where they meet and discuss anything. That didn't exist. So there is a real problem creating partnerships. Here, if you want to do this kind of research, for several years you have to work together, trying to understand each other. Now that is a big problem. And skepticism, prejudice is very strong in India. That has to be overcome. And obtaining financial support for this, because these are expensive. How do you get the money? So these are all the problems which we face. It took us some time to do this, and they were finally overcome. And I wrote this in 2006 towards Ayurvedic biology. This was Indian Academy of Sciences in Bangalore. At 2005, their annual meeting, I gave a talk in Benares. And the president at that time was Dr. Ramakrishnan, a very eminent physicist. So he asked me, can you please write this? We will publish it as a vision document. That is how it was published in 2006. I was not sure whether we would be able to do it, so I was cautious in saying towards Ayurvedic biology. Now, initially it received the support of Dr. Chidambaram. It is interesting that even though this document came out, It was seen by health department, DBT, DST, everybody saw, but nobody was willing to support. Everybody will say it's an excellent idea. But the person who really understood and gave the support was a neutron physicist, Dr. Chidambaram. I have incidentally seen this again and again. If you want to see really open mind in science, willing to look at even philosophy, you will find a physicist doing this. When I was developing a heart valve, for example, in Trivandrum, it's not easy to develop a heart valve because it has to beat 100,000 times a day, 36 million times a year, and the international standards say it should function without mechanical failure for 10 years. You have to demonstrate 360 million times without a mechanical failure in a wear tester. So it's not easy to do it. At that time, physicians condemned the whole thing. They said this is nonsense, ridiculous. Americans can do it. British are not doing. How can you develop this here? Nobody supported. No biologist supported. The only people who came forward, physicists, was Ramaseshan, for example. I can remember a few people. Why not? Engineers. These were the two pillars. Here again, here is something totally unconventional, and it was Dr. Chidambaram who supported this. And thanks to that, we had, these were the initial projects, which one is the genomic variation analysis of human dosha prakriti, which is very fundamental, vata, pitta, kapha. Is there a genomic basis for this? Studies on the biological effects of Ayurvedic formulations, that is, we chose Amalaki Rasayana on Drosophila, and studies on the effects of Amalaki Rasayana on the genomic stability of neurons and astrocytes in the brain. That was done by Dr. Kalluri Subbarao in Hyderabad. And physicochemical properties of Ayurvedic metal-based drug, RASASINDURA. This was done by BARC in Bombay and Ayurvedishala Kottakkal. And the immunological metabolic effects of Panchakarma at the GS Medical College and KEM Hospital, Mumbai, R.A. Podar Hospital, and ACTREC, Atomic Energy. So these were the projects that we started, a few projects supported by PSA's office. I'll just, I'm not going into details of all this, I'll just give the results of this so that you have a flavor of what is the type of research that Ayurvedic biology does. That is all my purpose here. I'm not an expert in these areas, but I'll go quickly through this. For example, this task force in Ayurvedic biology, after the only 3 years, Chidambaram's PSA's office could give the support. Then they stopped. So we had done enough work by that time. Published papers had come. Number of good institutions were collaborating. Vaidyas and scientists cooperating to do the work. So the DST was sufficiently encouraged. They took it over under the Science and Engineering Research Board. So they set up a task force in Ayurvedic biology. This is the successor to the earlier program. Here too, I am only presenting those where papers have been published. This is on can Amalaki Rasayana attenuate cardiac dysfunction associated with cardiac failure and aging. This was done by Rajiv Gandhi Institute in Trivandrum, evaluating the potential of Ayurvedic Amalaki Rasayana and RASASINDURA in suppressing Alzheimer's and Parkinson's disease. This is done in CCMB, Hyderabad. So these are the projects where papers have been published. I'll just go through this. Dosha Prakriti, the first one. Now, Vata, Pitta, Kapha, Prakritis, this is a fundamental concept. It is set at the time of conception itself. It doesn't change. And that determines the predisposition to diseases. It determines the individual response to treatment. So without knowing that, a physician cannot treat patients. But the question is, in the old days, it was determined on the basis of physical, mental, and behavioral traits. Based on that, they would say this is Vata, Pitta. But is there a biological evidence for this? That's the question today, which could not be done 40 years ago. But today we can do it. So this is, in those days, it was intuitively a physician would determine. It is still being done like that. We wanted to make it a little more rigorous, so the AYUSOFT program developed in this city, we used AYUSOFT also. Those intuitively determined, those same subjects would be seen by a IUSOFT expert who would use a computer-assisted determination. And we took only when there was agreement. And surprisingly, we had no idea whether there would be any agreement at all, but we have found that there is an 80% agreement. So only those subjects were taken for this study, so made it a little more rigorous. And 300 subjects in Udupi, 300 in Pune, and 300 in Bangalore, with the cooperation from Kalpana Joshi is sitting here. She was part of this in Pune. So the subjects were taken, and blood samples were taken from where there was agreement. And we chose only 2 markers. There are a number of markers we have to choose. So our molecular biology colleagues, they decided 2 single nucleotide polymorphism, SNPs, which is very widely used. Another is epigenetic alterations that are responsible for phenotypic differences. Epigenetics is almost Nature scientific reports. The principal work was done in CCMB, Hyderabad. They did this work based on SNPs. And they found that, you can read this very well, 262 well-classified individuals out of this total of 3,400 or so. And 52 SNPs were different between these prakritis without any compounding factors, because caste doesn't come into this, language, so many other things can vitiate your sample. So excluding all those, they came to this 262 well-classified people, and SNPs, they found 52, which were specific to these. And they, using these SNPs, 62, they were classified into groups of Vata, Pitta, and Kapha. And they also found there is a gene, PGMI1 gene, which we can see here correlated with Pitta phenotype. That's very important, because this gene is at the center of many metabolic pathways, which all have to deal with metabolism, like glycolysis, galactose metabolism, and so on. And Pitta essentially is also involved in these same metabolic activities. So there is, we cannot say that this is equals to that, but there is a very strong association. Now this was a paper, and subsequently others also have come. Now this was the DNA methylation for the epigenetic changes. This was done by Satyamurthy's group. Same blood samples were used. He was doing the samples were subjected to methylated DNA immune precipitation test. And there you can see the differential DNA methylation signatures in 3 distinct Prakriti phenotypes that demonstrate the epigenetic basis of Vata, Pitta, and Kapha. So this was also published in a top journal. Here is now biological evidence for these dosha prakritis. Then we move to the other project, microstructure or RASASINDURA, because it has always been a puzzle. Mercury, as you know, is banned. It cannot be used even for dental amalgam anymore. It's very dangerous, highly toxic. And in my student days, I can remember there was no diuretic at that time, oral diuretic. Only injectable, mercurial diuretics. Everybody was scared. You have to admit the patient when you give mercurial diuretics. Somebody coming with cardiac failure, swelling in the legs, you have to give diuretic. And because the renal toxicity was a very great danger. So it has been banned. But here, if you talk to senior Ayurvedic physicians, including the revered P.K. Warrior or Kottakkal, I have asked him myself. And they tell you, we don't use it often. But I have used it many times. I have not seen this problem. So you talk to Siddha physicians in Tamil Nadu, they will tell you the same thing. So there is a puzzle here. Instead of dismissing this as nonsense, that is not science. You have to test it. Now how to, any kind of chemical testing, mercury is there. So the objection applies. Then the question comes, can you look at the structure, microstructure? Now that is what this study, which Atomic Energy BRC did that, using X-ray absorption, fine structure. They have all the equipment here, perhaps the only place which has that equipment. And this analysis, the RASASINDURA was made in Kottakkal exactly as per the original protocol. and it is a single phase alpha mercury sulfide in their analysis. Unstable metallic mercury or beta mercury sulfide, they are not there. It is only pure alpha crystalline form. And Rasasindura is found to be comprised of nanoparticles of 24 nanometers, having hardly any free mercury or organic molecules. Rasasindura has a robust structure, negligible porosity. These are all taken from their paper. The non-existence of free mercury and the absence of unstable forms, robust character would ensure the integrity, because there is very little porosity, it is very stable. It will ensure the integrity of the drug during delivery and the prevention of its reduction to free mercury. And this places, this is a quote from their paper, this places Ayurvedic synthesis method at par with contemporary techniques of nanoparticle synthesis. And this is of course not given to patients, they have only done the structural study. But that is, this was published in Journal of Synchrotron Radiation, one of their top journals. And after this was published, that Devadatta Lahiri, who was the chief scientist, she was invited to the big pharma congress in the United States within 3 or 4 weeks to present a paper there, because they are interested in the toxicity of nano drugs and the delivery. Now here they found something in a nano form has been used for thousands of years. So there must be something they wanted to learn about the non-toxicity of this. So it is an important paper. Then immunologic and metabolic responses to Basti in obesity. This was done in Podar Hospital, and all the immunologic studies were done by Chiplunker's group in ACTREC. 32 obese individuals, they underwent this Basti treatment. There was significant reduction in weight, BMI, which was not our primary interest, but all those they have given. But the most important finding, the significant decrease in the production of interferon gamma, interleukin 6. These are the two very important. There is something else, an experimental, I won't go into that. A significant correlation, there is a tandem relationship between IL-6 and IL-8 at S2. S2 is after the treatment has started, S3 is 90 days later. And positive correlation between IgG and IL-6. And Basti modulates, the conclusion is this, it modulates the immune response by insulin resistance causing pro-inflammatory cytokines. And insulin resistance, as you know, the obesity, that fat is not just a passive lump of fat sitting there. It is actually highly active, chemically very active. It is producing a whole lot of things. Now these insulin resistance causing, these cytokines are all decreased, cut down by this enema, Basti. It starts from the start of treatment, S2 is after the treatment has started. And the effect continues to remain even 3 months after the treatment is over. That is the work which they had done. Now we come to Amalaki Rasayana, which was another project we took. And Rasayana is a general Rasayana, the whole body, as I indicated earlier. And this was done by, in fact, when the Rasayana project was being discussed, Kottakkal, our colleagues, they had wanted Narasimha Rasayana used. But Professor R.H. Singh, who is a member of our task force, he said, you know, why don't you use Amalaki Rasayana? It was his suggestion. So we accepted that. All this work, now there is a considerable amount of work on Amalaki Rasayana, which is very interesting. Now that was given, one of the first experiments done was Professor Kalluri Subbarao in Hyderabad. He had been working for many years on DNA chain breaks in the brain, single chain, double chain breaks. He was interested in that, its progression, its rate of repair, the way of detection, etc. So I asked him at that time, can you try this, our Amalaki Rasayana in the... He said, oh, there is no problem. That work which was done, it showed the DNA damage indicated by breaks in single and double chain. He would measure at 3, 9 and 15 months in all his experiments. That was done here also. And single and double strand breaks were significantly less in Amalaki Rasayana fed rat's brain compared to controls. This is all published in a very good journal. And both neurons and astrocytes in the Rasayana fed rats show greater genomic stability. Beneficial effect of Amalaki Rasayana on maintaining genomic stability in brain cells demonstrated clearly. This was the first paper which came. Now this was seen by Dr. Lakhotia, who is a very eminent Drosophila geneticist in Benares. He asked me at that time, see, this is a very good way science grows. Somebody has done work, it is published, you get a cue from that. That's where it goes. So it is one of the important ways science grows. So Lakhotia said, do you think I can try it in my flies? I said, there is no harm. After all, even flies, we share so many genes. There is no reason why it should not work. Why don't you try? So he tried. He was very skeptical. Initially, for example, the Amalaki Rasayana, the final stage, they use ghee and honey. So he said, maybe that is working. How do you know? So he tried a set of experiments initially giving only honey and ghee. Actually, that was bad for the flies. So it took 6 months to prove this to his satisfaction. But anyway, time was not important for us. So that work was done. And this was published in PLOS One. This is, of course, this I have already finished, Subbarao's. This is Drosophila. We have moved to the next. Now he found a series of findings in that the larvae fed on the Amalaki Rasayana showed earlier pupation and earlier adult eclosion. They grow faster. The salivary gland size greatly enlarged, enhanced levels of heterogeneous nuclear ribonucleoproteins in the DNA, significant increase in the total and hatched eggs in the female flies, their fecundity improves, medium lifespan is increased significantly. These are all findings in the Amalaki supplemented diet in flies. The thermal stress, Lakhotia has done a lot of work on heat shock protein, so he's an expert in this. Thermal stress tolerated much better. Starvation is tolerated much better. These are all the findings from that paper, which was published in PLOS One. It was not easy to publish. 3 times they sent it back. I was telling Dr. Thilu yesterday, first time a referee wrote back, we had given how the Amalaki Rasayana is made. That is cortical, the procedure which they follow. And we had given a reference of Ashtanga on Amalaki Rasayana. So the referee wrote back. That also shows the thoroughness with which they do this. A referee's letter, one of the referees, he wrote, this particular reference doesn't give all these things which you are mentioning. What we had described under materials and methods, that is not mentioned here. How do you explain this? Now the problem for us, Lakhotia was very upset because his molecular part there was no question, it was all accepted. Here is something they are asking about Ayurvedic reference. So when we asked cortical, they are very methodical people. They said, doctor, are you reasonable? See, here to make it, you pick 20 Amalaki. At the right season, you pick it, you dry it, you powder it, you mix it with an equal amount of fresh juice of Amalaki. You dry it, you repeat it 20 times. All these are there. You expect a book written 1600 years ago to give all these details and survive even today. And there are thousands of formulations. Is it conceivable? Are you reasonable? Now, it is not reasonable, but my problem is how to convince that referee. Otherwise, he won't accept. I mean, he's right also. So finally, we have to invent our own way of overcoming this, being truthful at the same time. So we resolved it by saying, materials and methods, we will only write what cortical has done, exactly. They can come and verify it. Their log books are there, no problem. But don't give any reference there. In the discussion, when you write, this formulation was made based on an ancient description. You give the reference there. Nothing. This was accepted. The referee was satisfied. So we have to be inventive even in the publication of papers because this is a new area. Now Lakhotia was sufficiently confident when this paper was published. In fact, it came within the PLOS One. They wrote saying that that particular year, the top 100 cited papers, this was one of them. So he was very happy. And he is also an expert on genetic engineering of Drosophila. human diseases can be induced in the Drosophila. Several. One is the Huntington's chorea, is a neurodegenerative, brain degenerative disorder. Another is Alzheimer's. So he had flies with these models of human disease. He gave it to them, and there he found it suppressed the neurodegeneration during the larval period without side effects, prevented the accumulation of inclusion bodies and heat shock proteins, suppressed apoptosis, elevated the levels of heterogeneous nuclear ribonucleic proteins, and improved the ubiquitin proteasomal system as a scavenging system that is improved. So this paper is also published, which has received, this has induced another scientist to do similar work. I will come to that. Now, this is the scientist working in CCMB Hyderabad. He had seen this work of Lacotia in flies. He had a mouse model, Dr. Patil, mouse model of Alzheimer's, chemically induced Alzheimer's, not genetically altered. Now he wanted to try this in his, that is only in flies, this is in mice. It's very different in significance. And there he found the effect of Amalaki Rasayana in ameliorating the memory and neurometabolism in the mouse model of Alzheimer's, chemically induced, compared with donepezil, which is a standard drug used, not very great, but that is a medicine which the FDA has approved in treating Alzheimer's. This is not a comparative trial. They were only trying to see, we know the mechanism of donepezil working. Is the mechanism of working of this Amalaki Rasayana in any way similar to that? This is all they are trying to. The question is that, not a comparative trial. And the AR treatment showed improvement in memory. The MACE test was done. This paper is published, you can read it. And it showed increased 13C labeling of amino acids, suggesting significant enhancement of glutaminergic and GABAergic metabolic activity in the mouse model. The above changes are similar to those produced by donepezil. So that current chemotherapy which is being used, the mechanism of action is very similar to what you see with this. This is a outgrowth of Lacotia's work. You can see how it goes. The traditional Indian, this is the most recent. Dr. Karta is an old colleague of mine. He works in Rajiv Gandhi, and he has been working for several years on cardiac hypertrophy. So if you have an obstruction, for example, aortic valve, there is an obstruction, or aorta itself, there is a coarctation, a narrowing, then the left ventricle has to pump much, much harder against this resistance. So it will keep on thickening. The muscle becomes thicker and thicker. Until at last a time comes, because when it becomes very thick, it cannot relax. Because heart, there is a systole, a contraction, diastole, a relaxation. So when the muscle becomes very thick, it cannot relax. And failure of relaxation leads to failure. That is true for all of us. If we don't know how to relax, then surely we are going to fail. So here, that cardiac failure induced by hypertrophy and cardiac failure induced by aging process, degenerative changes. In these, can this Rasayana do anything? That was the experiment which he did. And that AR intake improved the cardiac function, decreased LVH, left ventricular hypertrophy, increased fatigue time in treadmill test, in aging. It's amazing the kind of instrumentation we have today. In these rats, you can do treadmill test. Everything is measured. The size, the thickness of the left ventricle, the echocardiography, you can measure. I am amazed at the kind of instrumentation we have today, accurate measurements. And AR intake produced beneficial effect on myocardial energetics, muscle contractile function, and exercise tolerance. This is what I mentioned earlier. That first part we had actually shown, but they would not accept. So it took almost 2 years to do all these molecular level tests, but finally it got accepted. Now the current status of this Ayurvedic biology, it has been taken over as a task force in the Department of Science and Technology. It's funded by the Science and Engineering Research Board. It is appearing on their website. It supports research in modern science based on cues from Ayurveda. Funded projects from institutions all over India. Universities beginning to show interest in organizing courses at master's level in Ayurvedic biology. I think you are also showing interest here in Bangalore and somebody else. So there is a growing awareness of this new area. Young students wanting to work in this area. So that is encouraging. Now the scientific prospects here, really based on ideas. Where do we get the ideas for these? One of them is the study of classical texts. Now this requires joint effort by Ayurvedic physicians, Ayurvedic scholars. A physician need not be a scholar. We need an Ayurvedic physician who is also a scholar. So we need interaction between a person like that and a scientist. It cannot be done in just a meeting. If you are interested in any Ayurvedic problem, it may be Rasayana, it may be Rudacharya, it could be anything. If you are interested in that, repeated discussions, reading. Over a period of, it should be a small group, a study group, maybe 4 or 5 people, not a very large number. They share that interest. It may be in Rasayana, anything else. But they should be meeting, reading, and slowly some idea will come. Like this. After all, whoever thought we can show this effect, cardiac failure. At the very basic level, we can show changes. This has taken 4 years for the experimental work. A lot of discussion. I have to convince them, Karta and his colleagues, why would they want to use their laboratory facilities for a study like this? And they can do their own conventional work on coarctation. But that requires patience, discussion. For example, chronobiology, your subject of your symposium here. In chronobiology, suppose you want to do some work. How do you do it in the laboratory? If you want to do that, for example, Ayurveda says the 2 months, that is how it is decided, a year. But for convenience, they divide it into 2 halves, adana and visarga. Adana, all the moisture is taken away from the body, human body, from plants, from animals. We all become dehydrated, tired. That is one. The whole earth is dried up. But then the next half, all the moisture comes back. So everything is changed. This cycle goes on. But during these 2 halves, the important thing is, apart from all this tiredness and all that, the greenery is going. The chemistry changes, rasa changes. The 3 rasas are dominating in adana. The 3 others are dominating in visarga. Now rasa, remember, is the ancient term for chemistry. I already mentioned that. So what they are saying, Ayurveda, the body chemistry, body fluids, chemistry is changing. This is what it says. Now, here is something very interesting. If you look at chemistry today, we will look at electrolytes. We have various things. We do all that, and you cannot find any change because science today says there is internal constant environment. That's a fundamental principle. A dead body has no constant, it is equilibrates in the environment. But a living body, our internal chemistry is entirely different from external. Temperature is different, pH is different. That difference is life. This is Claude Bernard's very great physiologist, French physiologist. So we will not find electrolytes, you will not find. So you may say, oh, this is all nonsense. But if you have to do research in this is only in the last 48 hours since your chronobiology I am thinking. Now there it is possible. There may be something beyond chemistry, these electrolytes. When we were students, I give an example. Digestion in the stomach or gut is all based on enzymes. Saliva has enzymes. Gastric juice has enzymes. Intestinal juice has enzymes. Pancreas, enzymes. So these enzymes were digestion. That was for us. That was it. We had to know all that. Food is digested. They are absorbed, assimilated. That's it. We never realized many things we could not fully explain because there are people who are pure vegetarians. They are in perfectly good health. Where is their protein? You calculate all that. But they are quite healthy. Many things like this. But today, as soon as microbiome was known, now we realize we cannot say digestion is all because of enzymes. There is a whole new change in this, the way we look at it. So similarly, when you say these 2 halves, there is a difference. It is not difference in the electrolytes. Could be something else. Now, can we look for that? Now, here is a fundamental issue. It requires a lot of patience, a lot of discussion, a lot of work. But that sort of thing can be done in the lab. Now, this is really the study of classics. That is one way new ideas will come. Because these classics have survived for at least 2000 years. Charaka redacted it in 1st century. He says at the end of every chapter, Agnivesha Tantra Punasabskrite, I am redacting Agnivesha Tantra, which existed for hundreds of years earlier. So it's that old. So it has survived all these years. So we should look at it again and again. And these ideas are actually, according to me, children of God. Ideas. We cannot create anything. But you have to keep on trying. It's not easy to get children of God. You have to approach with respect, with humility, and when you get that idea, you have to nurture it. You cannot quickly do an experiment and say, oh, there is nothing in it. That's not the way to treat a child. Even if the child makes a mistake, you keep on trying again. That way you will get somewhere. The second is clinical experience of very senior physicians. Nowadays, all these half-baked, undocumented, many people, even our task force, we get reports like this. This is effective in something. Don't accept such things. We can accept, as far as I am concerned, aptas in Ayurveda, the source of knowledge, as you know. One is pratyaksha, what you see, what you observe. Another is anumana, inference. If there is smoke, there must be fire, that kind of inference. And the third, Ayurveda also accepts apta. That is the word of apta. Apta is a very exceptional type of person. He's a saint, he's an expert in theory, in practice, incapable of telling lies, who can see the present, past and future. Extraordinary individual. Such a person tells you something. You accept it. It is as good as paper published in Nature. For me, if, say, P.K. Warrior, 90 years in Kottakkal, he tells me, in this disease, if you give this, I think this can create this problem. For me, it is as good as reading a top journal, because that is source of knowledge, valid knowledge. If he says, don't do this, you better listen to him. Otherwise, you are going to learn yourself by causing harm to a patient. So that also is important, a source of knowledge. So a study of classical text, the other is clinical experience. Either it should be from aptas like this, I'll accept it. I'm willing to do research on that. Or it should be documented. Give me a report. You have treated 20 patients. These are the patients. This is what I did. This is the result. Show me that. This is never given. Don't accept that. Any project coming to us with undog, we just throw it out. We don't accept it. But this clinical experience, which is acceptable, that is another source of ideas. Cues from published work, which is what is happening here. Subbarao did some work, so Lakhotia does it, Lakhotia does, somebody else does it, so that grows. That is what is happening. Now, a university has begun an MSc course in Ayurvedic Biology, that is the institutional prospects with a holistic approach. This in Bangalore, and centers of excellence in Ayurvedic Biology. I see 2 or 3 universities are showing interest, setting up regular Ayurvedic Biology centers. Because one of the problems is people who have done this outstanding work, publishing in Nature and so on, when they finish their PhD, they have no jobs. So 3 of them, they have gone to pharmaceutical companies, doing work which has nothing to do with what they did earlier. One has become a teacher. So we are losing all these people. We cannot build this discipline. So there is an interest on the part of universities to set up centers of excellence. Investigators, there should be scientists with serious interest in investigating this, not just because they're transactional. There is a grant coming, you do the work and then forget it. That won't work. These are scientists with commitment. Similarly, Ayurvedic physicians are open to this. Without this, there can be no progress. And the support for PhD programs, Ayush has, as you know, established that. That is not enough. Our science departments, DBT, ICMR, DST, etc., they should offer PhD fellowships with contingencies for universities, postdoc career development awards, because after PhD, they are again stuck. As I mentioned, they have no job. So for 5 years, there should be a career development award where they can go, they get their grant, they get contingency grants, they can go to a university or an institute and work there for 5 years, during which they get their own grants, and by the end of that, there is no guarantee they will get a job there, but they will be wanted by institutions to start up their own. We are trying for this, and institutes and universities should be willing to offer opportunities for doctoral and postdoc work. If somebody comes like that with a grant, universities and institutes should create facilities, make it good for them to work there. It is a national investment that we are making. That is the current status of this discipline. I will stop there.